Welcome to What Your GP Doesn't Tell You, the podcast for both doctors and patients with me, Liz Tucker. This week, I'm talking to scientist Professor Sarah Berry about her work in personalised nutrition. This uses large numbers of research subjects along with various pieces of novel tech, such as continuous glucose monitors and remote testing, to gather huge amounts of data. This enables Sarah and the team who work with her to start to uncover the extremely varied effects food can have on one particular group or individual compared to others. This variability may be due to our genetics, metabolism, or indeed many other factors. Up to now, we've only been able to get very generalised population-level information, but this individualised research is revealing that our highly personalised responses to food may well have important implications for what, when, and how we should eat in the future. For example, research is revealing that as we age, we tend to have a worse tolerance for eating carbohydrates in the evening compared to earlier in the day. But women who take HRT can limit or prevent this effect. But before we get to the details of Sarah's interview, a brief request from me. If you enjoy this podcast and would like to leave a review on Spotify or Apple, that would be much appreciated. It really helps. You can also become a paid supporter of the podcast at patreon.com/whatyourgpdoesn'ttellyu or via PayPal on my website, whatyourgpdoesn'ttellyu.com. A huge amount of work goes into both the research and production of this podcast, so even a small amount of money makes a huge difference. And you can find out more information about the pod on my website, where you can sign up for the podcast mailing list, follow me on Twitter at Liz C. Tucker, and on my Substack account, liz.tucker.substack.com. Many thanks. And now back to Sarah's interview. Professor Sarah Berry is a nutrition scientist based at King's College, London University. She has particular research interests in individualised nutrition, food and fat structure, and the metabolic changes that happen after eating. She's also the chief scientist in the nutrition company Zoe. Here's Sarah's interview. Hi, Sarah. Thanks so much for joining the podcast today.
A pleasure. Thank you for having me on.
Well, I think, Sarah, for a long time, medicine's relied on large-scale trials and studies on a wide population level, and that's driven treatment. But in a sense, personalised nutrition's the reverse of this. Instead, we're looking at highly individualised approaches.
Yeah, absolutely. And I think that when we think about personalisation, whether it's personalised medicine, personalised nutrition, I think we need to think about multiple features of personalisation and bear in mind that we are already doing some of it. So we need to think about personalisation according to who we are, so our biology. We need to think about personalisation according to, in the field of nutrition, what we eat. We need to think about it in terms of how we live our lives, so what our lifestyles are. As our understanding of how diet or lifestyle or medicines impact our health. Now that we're getting to the stage of understanding, okay, this does impact our health, now we can go to that level deeper. And that level deeper is, who does it impact the most? How does it impact them? Who has the biggest benefit? Who gets no benefit? And I think this is a really exciting area of research because we see that there's huge variability. You have your responders, your non-responders. And I think that's why moving beyond the average, which we see is quite meaningless for many individuals, is really exciting.
But I wonder if we've had a sort of over-exaggerated view of how effective many population results are. I just remember a paper I read a while back in Nature that looked at the top 10 grossing drugs in the US, and they helped between 1 in 4, I think, and 1 in 25 patients, which I think to many people would be surprising.
Yeah, absolutely. I mean, that surprises me. But I know, having worked in nutrition, that there's this variable response. And we're seeing, for example, a lot of time invested looking at the GLP agonists, like Ozempic, who responds really well, who doesn't.
These are all the weight loss drugs.
Yes. So the weight loss drugs that are out there now. What we're starting to do with research, and this is really important when we're looking at the effectiveness of drugs as well, is to look at each and every individual's response to work out what proportion will get a benefit. So giving the kind of figures that you've said, what proportion will get actually a worsening of a given symptom? What proportion of people have no change? Now, what you might see is that 1 in 10 people, for example, get a benefit, but it's such a huge benefit that what that does is it pulls up the average so that when you present the average, on average, there's an X% improvement in X, Y, or Z symptoms. But I think it's looking at that proportion that get a benefit and looking at what proportion also may see a worsening of symptoms is very important.
And Sarah, of course, we should declare that you've got a conflict of interest because as well as being an academic, you're also the chief scientist for the nutrition company Zoe that does a lot of research in this area.
Yes, our main focus of research is looking at how diet and lifestyle impacts health. And yes, I do have a dual role. I'm always very open to declaring that. It's really important as scientists we declare all of the roles that we have. My primary role is as an academic at King's College London, where I've been an academic in the Department of Nutritional Sciences for the last 20 years. But I have also the exciting opportunity of working with Zoe as the chief scientist to, for the first time, actually translate scientific advice into actionable advice and also communicate it through podcasts like this, through our own science and nutrition podcast that we have at Zoe. I look back and I kind of almost feel like, wow, was I doing science for the sake of science? Because you get so engrossed in your narrow area of research. Yes, while I have this conflict, it's given me an amazing platform to be able to deliver advice using all of our new understandings out to a broader community.
So talking to a much wider group than you would have been able to talk to before.
Yeah, absolutely. In scientific research, the way it's worked in my field is you apply for grant, you spend years applying for grant, hoping you'll get it. 1 in 10 are successful. If you're successful, it then takes a year maybe for contracts, recruiting staff. You then do your clinical trial. So most of my research involves clinical trials, looking at different foods and how it impacts different health outcomes. Then eventually you write it up. From having the idea to writing it into a peer review manuscript, like a British medical journal, Lancet style manuscript. Well, we can aspire to those. You're talking anywhere between 5, 6, 7, 8 years, but then actually getting it out there to the general public in a way that can be interpreted and is relevant to people's lifestyle and can have health impacts is a whole other story.
But it's not just getting it out to the general public, is it? The time from a paper being published to it becoming accepted medical practice also has quite a long lag time.
Yeah, I don't work in a clinical setting, but from what I understand as well, the level of evidence that's also needed for it to become accepted clinical practice as well is huge. So there's a long time lag as well in terms of not just having one study, but having multiple studies that are all supporting in the same direction.
How much is the whole research area of personalised medicine possible now due to the various bits of remote tech that people can use, like continuous glucose monitors?
Yeah, so I think this is a really exciting part of research, not just in medicine, but in all biological areas of research. So in the past, we've had to rely on either running very small scale, but very high precision studies. So the kind of randomized control trials where you might have 20 people, you do really in-depth testing, and you get all of these different measures. Now, that's great to look at kind of mechanistic work or to get an idea of why something's happening. But it doesn't allow us to look at how different people vary either day to day or between one person and the next. So we've either had to rely on that or we've had to do these huge epidemiological style studies, these huge population style studies, where we might get data from tens of thousands of people, but it's what we call very low resolution data. We might only be able to collect one blood sample, or we might only be able to ask a questionnaire. And so whilst they both have their value, what we want to do, the kind of sweet point, is to be able to collect data at a scale, at a breadth, at a depth and precision that we can do, but not using those old kind of either the epidemiology or small studies. And this is what's exciting. These novel technologies that we now have available to us are allowing us to start to do this. I still think we have a long way to go, but being able to do remote clinical testing in a kind of community style setting allows us to be able to study a huge amount of people, a huge breadth of people as well, because this is really important, looking at different ages, sexes, ethnicities, socio-demographic backgrounds, etc. But it also allows us to collect data at that kind of scale we've just never done before, but at a suitable precision. So using things like digital devices. So now there's so many devices out there that can monitor your heart rate, can monitor how many steps you've done, can monitor your heart rate variability or other important clinical measures. We can also use other home testing tools. So we can use remote clinical testing. So continuous glucose monitors are one example. There's also the ability to collect blood remotely that you can send off to lab and have metabolomic measures taken that can look at hundreds to thousands of different markers. We can also use apps. So digital apps are a fantastic way that, again, we can collect data on how people are feeling each day. How's their energy, their mood, how's their hunger, how's different symptoms, etc. And we can track as well, these kind of things. So people from the comfort of their armchair can collect a vast amount of data on themselves that we can feed back to them as scientists, but also that they can share with us, that we can really start to learn and untangle the complexity of different individuals.
Now, of course, continuous glucose monitors have been seen as controversial by some doctors who argue that these are tools for people with diabetes and shouldn't be used by the worried well.
Yeah, so I think there's a couple of points to pick up on that. I think one thing that I feel is important to set the record straight on is there are some people, and I haven't heard actually clinicians say this, but you see on social media people saying, oh, these companies offering continuous glucose monitors are taking them away from individuals that really need them, that have, are living with diabetes. Well, that's incorrect. We know that in the UK, for example, there's no shortage of supply of continuous glucose monitors from the manufacturers. It's a case of whether at an individual level, the clinician decides it's a suitable option for their patient. And so that's one myth that is rather frustrating. Continuous glucose monitors originally were licensed to be used for people who are living with diabetes in order to enable them to control their diabetes. We now understand that if you have extreme excursions in circulating blood glucose, which typically will come about from a very high refined carbohydrate type of meal, that if these are excessive and repeated day after day over a long period of time, that this is associated with an increased risk of chronic diseases such as cardiovascular disease and type 2 diabetes. It's an emerging area of research. We don't have all of the very long-term studies that we have for some other risk factors. And I think it's important to accept that. But the totality of the evidence that we have to date is suggestive that if we can minimize excessive excursions in glucose, we may be able to improve health both in the short term and the long term. But it's really important to acknowledge as well that excursions in blood glucoses, so peaks in blood glucose, it's a normal physiological response to eating a meal. We don't want anyone to become obsessive about it. We don't want people to start trying to flatline their glucose. But what we do know is that if people can reduce it within the more normal ranges, they also start to say they feel better as well.
Well, it's interesting, actually, because I've tried using the Freestyle Libre continuous glucose monitor, and I found it actually gave me pretty useful information. My blood sugar is in a healthy range anyway, and I think I eat a pretty healthy diet. I always cook from scratch. But I discovered that there were two foods that really had an impact. One was all bran, which was about the only processed food that I was eating.
Yeah.
And it has a net carb content of about 12 grams, so not that high. But whatever I did, if I exercised before or after breakfast, unlike any other breakfast food, it continually peaked my blood sugar into the pre-diabetic range. Why might that be?
Yes, I think it's important first is to say it doesn't matter if it peaked it into the pre-diabetic range. Some peaks are a normal part of how your glucose would change over the day.
But it was much higher than any other food.
Yeah.
So it wasn't just the usual undulations. It was markedly higher than anything else.
Yeah, and I think that what we find is that might peak your blood glucose. It might not peak someone else's. I mean, 12 grams is quite a low amount of carbohydrate to cause that kind of peak. But what we know is if the carbohydrate's very refined, we know that therefore it can cause a very rapid increase. And I think one of the problems with that isn't so much the peak that you're getting, is a lot of people then get a dip after in their glucose. So you have this rapid peak, insulin suddenly pumped out to try and bring that peak down. and then it actually brings it down too low, and you can have what people often call like a sugar crash, where they feel pretty rubbish. And we've done quite a bit of research actually looking at these sugar dips about how people feel, how it impacts their energy intake, how it impacts their alertness, their mood, etc. For example, what's happening with all bran is most likely because it's quite refined carbohydrate, even though it has fiber in it. It's also not got much protein, if I recall correctly, and much fat in it. And so if you're eating carbohydrate that's refined, so it's very kind of accessible and quickly digested, and you don't have either protein or fat with it, then you've got nothing to kind of delay the rate at which your stomach empties. We also know that some proteins and fat also impact other gut hormones that also can impact your glucose response as well. And so this is why, as nutritionists, we always talk about the importance of having healthy fats and healthy proteins with your carbohydrates, that it's really important not to have carbohydrates just on their own, not just because of worrying about a high blood glucose response, but because generally you might get that dip. You won't feel full for long enough, and then you'll tend to overeat. Either you'll eat your next meal sooner or overeat at your next meal. And so thinking about getting that mix of fats, proteins and carbs, and avoiding things where possible that say low fat, because generally low fat doesn't mean it's better for you. And that's that's a kind of another myth that's still out there, particularly in my generation. You know, I'm in my late 40s, where the whole low fat's bad for you, reduce your fats came about. And that myth still perpetuates now.
Yeah, no, absolutely. So I always have full fat milk with the all bran. And I remember reading a study about a group who had been tested eating cornflakes, and 80% of them had been pushed into the pre-diabetic range eating cornflakes. And I'd always thought, oh, well, all bran's got much more fibre in it. It's got less sugar. So I was genuinely surprised. I mean, it had a worse effect on my body than chocolate.
Yeah, but then chocolate, although it's got quite a lot of sugar,
it's got fat in it.
It's got quite a lot of fat. I'm not saying chocolate's a healthy food, the kind of chocolate you get typically in the supermarkets, but it's also a great example of how we can't be too glucose centric. And so if we take chocolate, for example, and how that impacts your blood glucose versus all bran, if we were only looking at blood glucose, then we would say, oh, chocolate's better for you. Well, obviously we know that that's not the case.
Yeah. The answer is not to have a bar of chocolate for my breakfast instead.
Absolutely not. But how we have to use these new technologies like continuous glucose monitors, not in isolation. And I think that's a great example is of where we have to, if we're using any novel technologies, we have to look at the big picture. We have to look at the full picture and not only use single metrics. Because this is one of the risks, I think, that's out there. There are lots of companies using continuous glucose monitors, which we use at Zoe, but we use it in combination with lots of other things. There's lots of companies just using continuous glucose monitors, or lots of companies just doing microbiome testing, or just doing some sort of fat testing. There's hundreds of pieces to the puzzle that explain why you respond in a particular way to all bran versus chocolate. And it's important we look at all of those pieces.
But I must say, I have stopped eating all bran and I have other breakfasts instead. And I don't think that's done any harm. So I have nuts and seeds with yogurt or something, or scrambled eggs, all of which seem to be healthy alternatives.
Yeah, there's lots of fantastic healthy breakfasts people would have. To start your day off with a healthy breakfast that's keeping you fuller for longer is a great start to the day. Breakfast accounts on average in the UK for 20 to 25% of our energy. It's one of the meals like snacks that are under our own control more so than lunches and dinners, which typically might be controlled by who else we're eating within the family or our lunchtime setting. But I think as well, when you have a breakfast that causes one of these big peaks in glucose or dips, quite often you can feel it as well. So, for example, I used to have very highly refined carbohydrates for breakfast, and then I wore a continuous glucose monitor. I could see why about an hour and a half after having these refined carbohydrates for breakfast. So I'd have white bread, or I'd have pan chocolates, or I would feel ravenous an hour and a half after. I'd be grumpy on my meetings because I'd need to eat. And I could see I was having this big dip that kind of nearly 2 hours after. I now have a breakfast that's got lots of protein, lots of fat, and that keeps me going now till lunchtime. And so this is an example where they can be useful. I wasn't using the continuous glucose monitor to pathologize anything. But even though I'm a nutrition scientist that's worked in nutrition science for 20 years, it was like, okay, look, this is why I'm feeling rubbish. Let's change breakfast. So sometimes it's more of a motivational tool.
Sure.
And I think that's how we need to see these often, these kind of digital tools.
But the other food that I was really surprised by was rice, and it kept my blood sugar elevated. Normally when you eat something, it goes back to normal within an hour or 2 hours. But with rice, it kept it up for ages. And every time I ate rice, it had that effect. And then I saw somebody online posting their continuous glucose monitor, and I saw exactly the same pattern with rice. So after, anyway, the 2 weeks of the glucose monitor, the two things I don't eat anymore are all bran and rice.
Yeah, so I think that the kind of worst culprits for causing really big peaks or dips in blood glucose are white rice, white bread, white pasta. White rice and white bread are the two worst ones. Some people can handle it fine. So some people have almost no peak, no dip.
Bread was fine.
Yeah, it's interesting how it's different foods for different people. But again, I would say if you like white rice, you don't need to totally avoid it. What you could do is reduce down the portion size of the rice and add in a lot more of the protein and fat that you might be having with it. So let's say you're having some sort of, I don't know, chicken curry.
Yeah.
You add more of the chicken, add more of the healthy oils that you might be cooking with the chicken. It still won't totally blunt it, but we're not aiming for that. We're aiming for a response that you just feel good as well about.
It was weird that it just kept it up for so long, but it wasn't just white rice. It was brown rice, black rice. I tried it with different rice at different times of the day and kept all the other variables the same because I was trying to be quite sort of scientific about it. And so after that, I thought, well, whatever you're trying, clearly your body doesn't respond brilliantly to rice. And there's enough other options.
Yeah, we've studied this now in 200,000 people, how their glucose responses change to different foods, how it differs between people, how it differs within a person day to day. But what I find most fascinating, as well as looking at the data, because I'm a scientist and I get excited by the data, is anecdotally hearing stories like this from people, where even when I think about mechanisms and what could be going on, it kind of sometimes defies certain mechanisms. And it just shows you how individualized our responses are to everything.
Yeah, absolutely. Now, one of the personalised nutrition studies you've done was called the Predict1 study. So what information were you collecting in that study?
Yeah, so this was the first study that we performed at Zoe, and this was a study really to answer 3 key questions. It was a study to look at how much variability there really is between individuals and how we respond to food, but also to have a look at how much variability there is within an individual day to day, because this is a really important area of personalisation that if we've got time, I'd love to touch on as well.
Yeah, absolutely.
So we can personalise based on how I might differ to you, but we can also personalise how you might differ day to day, depending on how you live your life. We also want to look at what's explaining these differences. So we've seen there's huge differences, but then we want to look at what explains these differences. So we collected lots and lots of information on what people were doing day to day, on what they were eating, their demographics, different phenotype information. So we were looking at, are you pre or post menopause, or what's your age, what's your sex, what's your microbiome like, looking at their genetics, etc. So that having seen how much variability there was, trying to explain that variability. And then the last thing was seeing, okay, can we then use all of this huge data to actually build prediction models so that we can apply this to the wider population? Can we predict how people might respond to food? So this is what we did in the first study, which was in 1,100 individuals, the majority of whom were twins from the Twins UK cohort, which is led by Professor Tim Spector, who's one of the science co-founders of Zoe.
Been on the podcast actually a while back.
And we collected a vast amount of information over a 2-week period where we leveraged all of these technologies that I talked about earlier. So continuous glucose monitoring, we did loads of finger prick testing for different circulating metabolites, both when people were fasted, having just got up, as well as after them having meals. We collected lots of information on their sleep, their activity, how they were feeling every few hours. What's your hunger like? What's your energy levels like? What's your mood like? And as well as lots of questionnaire data on what their typical diet is, their typical lifestyle is, etc. And then we also collected microbiome samples. so that we could feed all of this into what I think of as a black box, because we have incredibly clever data scientists that kind of blows my mind what they do with this, so that we could look at, firstly, how much variability there was, but how much of that variability is explained by different outcomes. And we saw that there was huge variability, even with identical twins, in the different outcomes that we looked at. But we could start to disentangle the pieces of the puzzle and what are the biggest pieces that are explaining this variability and what are the smallest pieces.
Well, should we look in a bit more detail on some of those markers you looked at?
Yeah.
One of which was triglycerides. Why are those important?
So when we think about triglycerides, I think we need to think about two different areas related to triglycerides. Firstly, we need to think about fasting triglycerides, and then we look at also postprandial triglycerides, which just means post-meal levels of circulating triglycerides. So when we look at fasting triglycerides, we know that that gives us a really great measure related to cardiovascular disease. And it's something that as scientific researchers were very interested in. We know that diet has a huge role to play in determining our triglyceride levels. Yes, we know there's some genetic factors and there's other factors related to our health, but we know that diet can modulate our fasting triglycerides. So we know, for example, if you have very high refined carbohydrates, that can increase our triglyceride levels, as well as having the wrong types of fats. So, for example, saturated fats as well, because they impact how our liver produces triglycerides. So triglycerides in the fasting state gives us a good measure of what I think of as our overall metabolic health. Triglycerides alongside glucose give us a great measure of our metabolic health. We know that fasting triglycerides are an independent risk factor for cardiovascular disease, independent to cholesterol, and it works through slightly different mechanisms. So that's one of the measures that we looked at. What are the fasting levels and how much variability is there in the fasting levels?
And Sarah, I think it's probably worth mentioning that when people get their lipid panels done from their GP, they're not told to fast beforehand. But actually, if you want to get an accurate measure of your triglyceride level, you should fast the night before you get the lipid panel done.
Absolutely. So we know that for cholesterol, so our HDL cholesterol, our LDL cholesterol, our total cholesterol, whether you fast or not has a minimal impact. It does impact some of the triglyceride content of the cholesterol particles. And if you were to go into a really complex lipid panel that we do in our studies, yes, it might have an impact. But actually for total HDL and LDL cholesterol, whether you're fasted or whether you've just had a high fat meal is almost irrelevant. Where it becomes really relevant is for your triglyceride levels. And the reason is because when you have a meal that contains fat, what happens is that fat circulates in your blood before it's removed from the blood in specialized particles in the form of triglycerides. So when you consume fat, 98% of the fat that we eat, regardless of whether it's the fat in olive oil or the fat in chocolate, it's in the form of triglycerides. So fasting triglycerides represents the fat that's coming out of the liver, that your liver's making. But in the post-meal state, so about 4 hours after having a high-fat meal, you have a big increase in circulating triglycerides that's come from the meal that you've eaten. That returns to baseline around 8 hours. So if you're consuming multiple meals throughout the day, you actually spend most of your day in this postprandial state, this post-meal state. So your triglycerides remain elevated pretty much throughout the day, except in the fasting state. And because we now know that triglycerides are a really important metric related to health, it makes a lot of sense to be advising patients, please do come in fasted for a lipid test so that you can also look at the triglyceride level alongside the cholesterol level as well.
So when you were looking at these triglyceride levels, what effect did genetics have on that?
So we looked at the impact that genetics had on the post-meal, so this postprandial change in triglycerides, as well as for postprandial glucose and inflammation, etc. And we compared one identical twin against another identical twin. And we actually simply plotted it to start with on a figure. So imagine a figure where you've got one identical twin on a y-axis and you've got the other identical twin on the x-axis. Now, if the trait was all due to genetics, you get a beautiful straight line going through the middle. But when we did that for triglycerides, the dots, so the individual values, were all over the place. And we could actually also use something called ACE heritability modeling. I'm not a geneticist, but it basically allows us to tease apart how much of the response is due to genetics, how much is due to people's upbringing, and how much is due to the environment. And you can do that if you have a kind of study like we did it in PREDICT, where we had identical twins, non-identical twins, and unrelated individuals. And what we found was for the postprandial triglyceride response, less than 2% seemed to be due to genetics when we did that modelling. Yet when we looked at the postprandial glucose response, about 45% was due to genetics, showing that genetics actually was playing a really small role. Now, this is exciting in one respect because it means that we potentially do have a lot of control over this because, I was a child brought up in the 70s, 80s, where it was the era of genetic discovery. And my mum would always say, oh, it's all in your genes anyway. And seeing this kind of data is quite exciting because, yes, we know there are some genetic conditions that will cause excessively high cholesterol or excessively high triglycerides. But for the majority of people to show actually it's not just due to our genetics, it's quite empowering because it means actually we can tell people, or clinicians can tell their patients, look, there is something that you might be able to do about this.
And what are the things you might be able to do about it when it comes to triglycerides?
So it's still an emerging area of evidence. To reduce your fasting triglycerides, one big factor can be reducing the amount of refined carbohydrates that you have. So reducing the amount of carbohydrate that's being delivered to the liver, that's then pumping out your triglycerides, increasing your activity levels, reducing your alcohol intake, eating more fiber, eating less ultra-processed food. There's also some evidence that increasing the amount of omega-3, so particularly from oily fish, omega-3 fatty acids, can also reduce your triglycerides as well.
And so within one individual, say within a week of my meal patterns, what sort of variability am I likely to see in my triglycerides?
So what you're likely to see over the day is huge variability because every time you eat a meal, what happens is the carbohydrate in the meal causes a short-term increase in circulating blood glucose that peaks about 30 minutes, returns to baseline around 2 hours. You also then have this increase in circulating triglycerides from the fat in the meal that peaks around 4 hours, returns to baseline around 8 hours. If you then imagine how we typically eat in the UK, on average, people have 3 meals, breakfast, lunch, and dinner. And on average, people have 2 to 3 snacks a day.
On average?
On average, yeah.
Wow.
In the UK, 25% of our energy comes from snacks. That is
terrifying.
Huge. But although it's huge, it's exciting because it means there's something we can do about, using one simple single strategy to change snacks. Anyway, so that means that if we're having about 5 to 6 eating events, as we call it, throughout the day, you can just imagine in your brain, think of these hills and these mountains, which are, these changes in circulating glucose, circulating triglycerides, but also you have loads of downstream metabolites that change. So we know that when this increase in triglycerides and the increase in glucose also causes an increase in inflammation. So there's certain inflammatory mediators that increase postprandial. And there's lots of other things that happen. You get an increase in hemostatic factors, so blood clotting factors, for example. And it's these increases in glucose and triglycerides that cause these downstream changes in circulating metabolites, which is why if it's repeated, excessive and uncontrolled over long periods of time, then goes on to cause problems.
Now, Sarah, you mentioned that when it comes to our blood sugar levels, genetics plays a much bigger role.
Yeah, so that's what the data from our PREDICT-1 study has shown. We would like to repeat this analysis and confirm this. But yeah, there does seem to be more of an impact of genetics in the postprandial glucose responses than there is in the postprandial triglyceride responses. Why? I don't know the answer to that yet.
But that still means, I think you said 45% was affected by, which still gives you 55% where you can take individual action.
Yeah, absolutely.
So what we're really trying to have is a blood sugar pattern where it's more sort of gentle undulations rather than lots of major peaks and troughs.
Yes, that would be an ideal world. But I think, again, and I can't repeat this enough, it's a normal physiological response to have some peaks, to have some troughs. And I think for anyone that is trying a continuous glucose monitor that isn't living with diabetes, to be mindful of that being a normal response and not to over worry, because that's the one thing that we do a lot at Zoe. We spend a lot of time educating people to what is a healthy response and not to worry with having, some foods, of course, are going to give you a peak, and don't worry about it. Having that every now and then is not a problem. It's if it's continuous throughout the day.
And did you find that some individuals who have a healthy blood sugar level have a greater tendency to greater peaks and troughs?
Yeah, so some people who have a very healthy fasting level may have a much higher peak to some meals compared to other individuals. And this is why it's a really interesting area to look at as a nutrition scientist, to look at these postprandial responses. What you often see is more of a differentiation in people. So you might see people who, in a group of, let's say, 100 people, they all have broadly similar fasting levels. So based on their fasting levels, whether it's glucose, inflammation, triglycerides, whatever, that actually they would all be grouped together in terms of the same level of risk. But once you then kind of challenge the system by giving either like a high fat or high carbohydrate meal, and then you look at those postprandial, those post-meal changes that happen in that 30 minutes, 4 or 5 hours after a meal, you start to see a greater differentiation in people's responses, with some people, despite having the same fasting levels, having really high postprandial responses, and some people having low postprandial responses when they're given exactly the same meal. And so the hypothesis from this is that actually maybe this is a really great idea for us to see early indications of some sort of metabolic disturbances that are happening, and maybe measuring people postprandially as well as fasting will allow us to discriminate risk better. And I think this is a kind of let's wait and see what more comes out of that.
So whether it's potentially an early red flag or not?
Yes, absolutely. I've spent, before I started working at Zoe, the previous 20 years looking at how food structure and processing impacts these postprandial responses, because we believe as nutritional scientists, it is something that firstly can be negatively impacted by food, but secondly could be considered that early red flag.
And time of day seems to have a major impact on our response to carbohydrate.
Yeah, so this is where I think it gets really exciting. So we've been able to look in our PREDICT study as what explains the variability between individuals. So we see, if we take glucose, for example, there's about a 20-fold variability in how different individuals respond to exactly the same meal. So you might respond 10 times greater response, or I might respond 20 times greater response to someone that's listening to this podcast. And we're able to look at, for the different outcomes, what is it that's explaining that variability, as well as seeing what explains how I might differ to you, Liz, whether it's partly my genetics, partly my age, partly my microbiome, partly some of my other kind of fasting blood measures. What we also know is that we vary day to day. So how you respond to a carbohydrate meal varies within the day due to time of day, but also day to day as well. And I think this is really exciting because, again, I think it gives different tools in our toolbox to be able to modify how we respond to food. So on average, what we see is if you consume carbohydrates in the morning, people on average have a lower postprandial glucose response compared to if they consume them later in the day. So people consuming carbohydrates later in the day tend to have a higher postprandial glucose response to exactly the same meal that they might have had in the morning compared to if they eat it in the morning. We see this differs for different people. So we see that, for example, that time of day effect, as we call it, is even greater in postmenopausal women. So if you're postmenopausal, then your blood glucose response to a carbohydrate meal is even greater in the afternoon compared to a premenopausal woman. And so, again, this is the kind of thing that if we think about, well, what does that mean practically? Well, what it could mean is if you are concerned about your blood glucose levels, but you like to have cake, maybe have it as your mid-morning break rather than your afternoon break. This kind of gives us some simple strategies that we can implement.
But I did see some work you did, Sarah, that suggested that as we age, our response to carb timing was less important?
So our initial study that we published did find that there seemed to be less of a difference in postprandial glucose responses as we've aged. That was some unpublished research. We've since done a deep dive of some new data we have, and this is from our PREDICT-3 study. This is a study where we have hundreds of thousands of people, and we've been able to break them apart a little bit better according to whether they're males or females, peri or postmenopausal. And we actually see the reverse of what we had seen in that original study. Once we start to break them down, then actually, depending on whether you're male or female, that as you age, this time of day effect seems to actually get bigger.
So the reverse of what the unpublished work first suggested.
Yes, which shows you just how complicated it is and how important it is to verify these results in different cohorts.
Absolutely. And I think it's true, isn't it, that generally speaking, our fasting blood sugar level goes up anyway as we get older.
Yeah, so this is something we've published about a year and a half ago in eBiomedicine, where we looked in our PREDICT-1 study at individuals' responses according to age, according to sex, and according to menopause status. So we're particularly interested in how menopause impacts our responses to food. And what we found was that as you age, whether you're male or female, your blood glucose responses and loads of other health outcomes, which I know you'll be more than aware of, blood pressure, cholesterol, adiposity, etc., glucose, it slowly goes up. The trajectory for men is slightly higher than for women up to the menopause. So men tend to be at any given age having slightly higher levels of cholesterol, blood pressure, glucose, etc. As soon as women hit the menopause, suddenly it jumps up and it can often overtake men.
That's interesting. And did you differentiate between women taking HRT and those not taking HRT?
Yes. So we looked at the whole cohort, and we also looked at those taking HRT, those not taking HRT. HRT blunted or even prevented, in many cases, this kind of jump that we saw. So in those taking HRT, they did not have the same increase in glucose and triglycerides at fasting or postprandial as well. They had significantly less of an increase in cholesterol, blood pressure, visceral adiposity, etc., which confirms what we're seeing now with other studies that are coming out.
Now, at the beginning of our interview, you touched on the fact that it's not just the carbohydrates. The important thing is the food that we eat our carbohydrates with.
Yeah.
And if we eat naked carbohydrates, we'll get a much greater response.
Yeah. So if you eat carbohydrates on their own, then you've got almost pure glucose or the carbohydrates going into your blood. If you have fiber, protein, or fat with your carbohydrates, it impacts your blood glucose response a number of ways. One, it can slow down the rate at which your stomach empties. So you don't have this sudden release of the glucose into your bloodstream, but it's slowing down how quickly it's coming out of your stomach and therefore entering your bloodstream. You also have different gut hormones that are released in regard to particularly protein, but also fat, that also impact insulin release and also some other gut hormones that also modulate your glucose. So it kind of slows it all down. It changes it from being this, massive kind of spike, although I am reluctant to use that word, but this spike into that more undulating hill, as you said. But it's not just what you're eating it with. The structure, so what we call the food matrix, is also really, really important. And this is where this whole area of ultra-processed food becomes really relevant as well.
Because with ultra-processed food, a lot of the food processing has already been done for us. So our bodies have less work to do in breaking down the food. And as a result, that pushes up our blood sugar levels.
Yeah, absolutely. So when we process food, lots of different things happen. And so what we know is if you change the structure of food, you change many different factors related to how your body is going to respond to that food. So firstly, we know that on average, people eat ultra-processed food about 40% more quickly than an unprocessed equivalent. So let's take an apple versus a pureed apple, for example. That pureed apple could be eaten about 40% more quickly than that whole apple. So you're actually, firstly, even delivering the carbohydrate to your stomach even more quickly as well. So you're increasing the rate that it's delivered. We know that if you increase your eating rate, we know that that increases your post-meal blood glucose response. It also increases how many calories you have. And this is a big problem, I think, that people don't often think about when it comes to ultra-processed food, is that you tend to eat them really, really quickly. And then you haven't got time for your fullness signals to go to your brain to say, hey, you've had enough, slow down, you don't need to eat anymore. You've already finished it before that's even got to your brain. And so that means that people on average consume more calories as well. And we know from a randomized clinical trial where people were fed nutritionally matched meals, but one was ultra processed, so the food structure was destroyed, versus those in the original structure. They consumed 200 calories more per day, even though they were nutritionally matched if they were ultra processed. So you're consuming more calories, you're eating the calories faster as well. It impacts also your feelings of fullness. It impacts also where the nutrients are absorbed. So what we know is if it's more heavily processed and the matrix is destroyed, it's absorbed higher up the gastrointestinal tract where you have fewer of these kind of fullness receptors as well. So there's lots of different things that are going on in relation to how that food is processed purely due to the food structure. Never mind, we know that with ultra-processed food, there's lots of additives and emulsifiers that we don't fully understand yet, how they impact our health and our microbiome. The fact that ultra-processed foods tend to be higher in sugar, saturated fat, salt, and lower in healthy oils and fibre as well.
Well, that's quite good news for me because I'm a slow eater. But the trouble is with my family, if it's any occasion where it's shared food, you can do very badly because unless you eat quite quickly, all the food's gone.
I know, it's really interesting because we've been doing some work on eating rate. And a common thing that people often say is, well, I come from a big family and it's kind of survival of the fittest, who could eat fastest to get their second portion. Or, even at my children's school, lots of schools have expanded. The canteens have remained the same size. So my children have like 10-minute slots where they go in and eat their meals.
10 minutes?
It depends whether it's packed lunch or whatever. But, they have 10 to 15-minute slot where they're in the dining room to quickly eat their meal. And they always say, yeah, we're told, hurry, hurry, hurry. Because at home, I'm like, slow it down. Research has shown that if you slow down the rate at which you eat your food by just 20%, you reduce your energy intake by 15%. Now, I know that's not relevant to everyone, Liz, but for those who are trying to maintain weight or lose weight,
yeah,
that's another tool that you could use. Just put your knife and fork down between each mouthful. The problem is, though, now we're not eating in a mindful way. Many of us are gobbling down our food while we're on a Zoom call or quickly going into a meeting, and we're not taking our time with our meals.
So food order is really important too. Does that mean have your salad before you have your main meal, for example, rather than with the main meal?
So there's lots of research coming out on this about what we call in science preloads, as in should you have a preload 30 minutes before of a fibre, so like a salad.
Or a bit of avocado or something.
Yes, rather than with your meal. I think that there isn't enough evidence to warrant changing how you eat your meal from having your salad with your meal versus a little bit before and the size effect that it might have. And I think this is really important actually to touch on, is that when we're thinking of any advice in terms of nutrition, in terms of how we're eating, and I'm sure for any clinicians listening, this is something that you think about with everything that you advise your patients. But something that we have to think about in nutrition science is the size effect. What's the size effect of eating that salad 30 minutes before versus eating it at the meal? Is that size effect big enough to warrant the potential change it might cause to the pleasure of the meal?
Because you still want eating food to be fun.
Yes, exactly. Food is there to give us joy. It's there to give us pleasure. It's there to taste great. It's part of our emotions. It's part of our culture. It's part of what defines for many people who we are. And we mustn't forget that. In the quest to eat healthily, we mustn't forget the pleasure of food. And this is so important. And so I think, yes, there's evidence to show if you have fibre, fat, protein prior to a meal as a preload, it will reduce your glucose response to that meal. There's also evidence to show if you add it into the meal, it reduces that glucose response. Whether having it 30 minutes before the meal or with the meal has a big size effect, I don't think there's enough evidence to say yet. What I certainly know is that I can't live my life where I go and have a salad and then wait 30 minutes to have the other component of my meal. Or I don't want to sit in a restaurant where I eat just one part of the meal that's on the plate and then wait 10 minutes and then eat the other part. I want to enjoy it. And I think this is where we have to be a little bit mindful. And also, we're only talking about one thing here, the blood glucose response. That's only one part of how we're responding to that meal. So yes, there is some evidence, but I would be a little bit cautious. And I would just make sure your meals are a mix of healthy proteins, healthy fats, have some carbohydrates if you enjoy them. And most importantly, try and make sure as much as possible these foods are not heavily processed, that they look like the original plant or food that they came from.
But if you are, for example, going to have a bit of cake, you will be better having it after a main meal than you would in the middle of the day when you haven't had anything else with it.
Yeah, so there's some evidence to show, yes, eating a high carbohydrate load on its own will cause a higher glucose response than if you have had in, let's say, the previous hour, some sort of meal that's containing fat, protein, or fiber.
And other people have suggested that spices such as cinnamon can have an impact.
Yeah, so there's so much evidence now coming out around herbs, spices, vinegars, these sorts of things. I think it's something that, yes, at high doses, things like cinnamon or turmeric or some of these other spices may have an impact. Maybe one of the reasons why some of these spices are shown to have long-term health benefits in terms of inflammation, for example, or inflammatory related diseases. I think the evidence isn't strong enough yet to support people taking these as kind of pre-meal capsules to say, hey, have a capsule of turmeric or something or cinnamon and it will blunt your blood glucose response. If you like the taste of it, absolutely add it to your meal. The more diversity that we can get in terms of plant-based foods and herbs and spices that have different chemicals that will all impact our health in different ways by giving us these bioactive compounds. So in nutrition, we no longer just think of food as fat, protein, fiber, carbohydrate. We realize that on average, every food has about 20,000 chemicals. And many of these have bioactive properties. They have these kind of almost like pharmacological-like properties that do wonders over periods of time, not typically at the level that you might add in a single sitting. I know some people say, oh, I take apple cider vinegar before, but I hate it, but it's going to lower my blood glucose. Well, if you don't like it and you don't have any evidence that it lowers your blood glucose, and actually given that blood glucose is just one of many, many, many things that we think about, that's where I would say, go back to that thing. Look, your food's to be enjoyed. Don't obsess.
Well, actually, when I had the continuous glucose monitor, I did try using the apple cider vinegar several times, and it seemed to have absolutely no impact whatsoever for me personally.
Yeah, and this is where I think we need to be really careful, because there's lots of influencers out there. There's lots of people on social media saying, hey, I did this and look what happened. Well, great. That worked for you, but what works for you doesn't work for everyone. And this is what I think we need to be very cautious about in personalized nutrition, for example. Whilst it's an incredibly exciting area of research, what works for one person won't necessarily work for another person.
And Sarah, one part of our body that seems to show huge variability between individuals is the gut microbiome.
Yeah, so the gut microbiome differs hugely from one person to the other, but it also differs day to day, month to month, depending on lots of factors. Have you taken antibiotics? What have you eaten? What have you drunk? What are you doing in your life at that moment as well? And we see from our Predict-1 study, but also this Predict-3 study, which is in 200,000 people, we see such an enormous variability. We see an enormous variability also even within identical twins, which again is really interesting because again, that shows how actually genetics plays a tiny role in shaping your microbiome. And we see that a lot of the variability in your microbiome can be explained by the foods that people are eating. And that actually you can change your microbiome in as little as 3 days. There is a kind of core underlying microbiome that's more difficult to shift. But one of my colleagues, Dr. Emily Leeming, has done some really fascinating work showing that actually after about 3 days of changing your diet, you can actually see a shift in your microbiome composition. The thing is you need to keep that going. And this is a problem with, some of these probiotic drinks that people take or probiotic capsules. people might say, oh, well, I'm going to take these drinks for a couple of weeks. It's going to improve my microbiome. Yes, it will. But actually, you then need to maintain it. It will just go back to how it was unless you're keeping that going.
Now, interestingly, I interviewed a dentist recently for the podcast, and she said there's not nearly enough attention given to the oral microbiome. And the oral microbiome really affects the gut microbiome. So if you're just going to look at one and not the other, you're missing a large part of the picture.
Yeah, so this is something I know very little about at the moment because there's actually very little studied on it. I think this is a exciting new area of research. And I know I've said this about so many things, Liz, but it's because there's just so much new research coming out. I think it's been really understudied, and I think that we'll start to be able to piece together how the oral microbiome is linked to our gut microbiome a lot better. You know, there's like the vaginal microbiome, there's the skin microbiome. We're starting to see all of it is so important. And again, it takes me back to what we talked about at the beginning about these novel technologies are going to give us this exciting opportunity to piece together these thousands of pieces of the puzzle.
But how much do we know if we are changing our microbiomes, in particular the gut microbiome? How much do we know that we are changing it for the better?
So what we do know is from what we call cross-sectional studies. So studies where you look at a single point in time and you look at measures of health, you look at measures of microbiome. We do know that there's certain species that are associated with certain measures of health, some favorable and some unfavorable. So for example, in our Predict1 study, we developed what we call this microbiome signature, where we saw a very clear cluster of species that were all associated with favorable measures of health. So lower blood pressure, lower inflammation, lower cardiovascular disease risk, lower glucose, lower cholesterol, etc. And then conversely, we saw this cluster of different species that were all heavily associated with unfavorable measures of health. So higher blood pressure, cholesterol, glucose, insulin, etc. Now, that gives us the first indication there's something interesting going on, but it doesn't show causality. And this is what's really, really important now in microbiome research, is for us to show that there's a causal relationship, i.e. those species are causally changing these health outcomes. This is where we now have new research emerging from either randomized controlled trials or also longitudinal data as well. So we've been doing a lot of work at Zoe looking at how changing someone's diet might modify the microbiome, so these different species, which might then have impacts on health. What our current results are showing is that if we change the diet, so we increase the healthiness of the diet, we see an increase in what we call these good species, the ones that are associated with these favorable measures of health. We now see that very clearly from some research that we've recently completed. It's not published yet, but it will be out very soon. The next step in this is to do that final link where in our data, we see that then this changes these health outcomes. Again, we need to do more research on this, but I think that the research that's coming out from other studies as well is really, really promising, that there are certain species that we know are favorable for health. Whether there's a universally classified a good microbiome and a bad microbiome, no, there isn't one yet. But I think that we're a long way along the path of developing that now.
So is what's healthy for you in your gut microbiome going to be what's healthy for me?
That's a really, really good question. It's not something I have the answer for. What we do know is that certain people have less favorable species, certain people have more unfavorable species. And what we do know is that by modifying the diet, using this data that we have of showing which foods are associated with which species, we know from our Zoe Predict research that we can increase these good species and reduce these bad species through these very personalized dietary modifications. And just in case there's any microbiologists listening, I'm using the term good and bad species in a very kind of loose setting.
Sure.
Because that might irritate them. I think it might be that different foods differentially help different bacteria, different species. But I think that the evidence that we currently have would show that there are some species that we believe are associated with healthy health outcomes in everyone, and there are some that are associated with unhealthy outcomes in everyone. What we need to start doing in microbiome research is looking at the functionality of the different bacteria. So at the moment, what we typically look at is what species are present, but we also know how they function is really important.
And we know that some people who've got diseases, like, for example, somebody typically with MS is likely to have a different gut microbiome than someone who doesn't have MS.
Yeah, and I think this, again, is where there's some really interesting data coming out. So if we look at colon cancer, for example, and we've just been awarded, both Tim and myself at King's College London, a huge grant from Cancer Research UK and other cancer charities called Prospect. This is a $25 million grant to work with international collaborators to look at early onset cancer, to look at assessing risk of early onset colorectal cancer and also prevention of it. What we know already, but we're going to dive into this more deeply as part of this grant, is there are microbiome signatures that are quite predictive, not with 100% accuracy, but are very predictive of people who may go on to develop or might be in the early stages of early onset colorectal cancer. I think that we'll see this emerge more for other diseases as well, but I think certainly for the colorectal cancer area, that's fascinating. And again, this opens whole new areas for screening in the future once we understand fully what's going on there.
The million-dollar question then being, if you can change those early signatures, whether you can prevent these diseases developing or perhaps prevent a less severe form of the disease.
Yeah, so this is what we're going to be doing with this Prospect grant. As part of it, we're going to be conducting a randomized control trial. This is being led by a colleague, Professor Jordana Bell now, at King's College London, looking at how we can modify this particular microbiome signature to see if that goes on to have a knock-on effect in reducing your risk of early onset colorectal cancer. So in 5 years, we will know a bit more.
Really, really interesting. And finally, so does it worry you that a lot of the people who are looking at these personalised nutrition approaches in terms of the public are the better off? Are we forgetting those who don't have the same resources? For example, people who couldn't afford a Zoe membership.
So I would answer that in 2 ways. I would answer it firstly as Sarah the scientist. As Sarah the scientist, what we're doing at Zoe, using the data from people who can afford it, is phenomenal in terms of the advancements we're going to make in our understanding of how food impacts our health, in the kind of data we're collecting that we can share with other scientists that can really push the fast forward button on our understanding of nutrition, lifestyle, and disease. So I think we need to be clear that there is that benefit that we can get for the greater good of society and science. Yes, it's a concern with any nutrition activities, with any lifestyle activities, that it's often people already have a healthy diet that become more aware of the importance of a healthy diet. And it's a huge problem in nutrition, the disparity that there is between, the top 5% of the population that are already eating healthily. You've got a proportion in the middle, and then 50% of the population who are not eating healthily because of many barriers. It's beyond a simple company like Zoe to ever be able to close that disparity, because it's such a huge problem. It's something that has to be addressed globally, but also if we think of the UK at a government level, it's a big problem, and I recognise that. I think what we can do at Zoe is, one, benefit, like I said, from the science, one, try and educate people. So we have our free Zoe Science Nutrition podcast. We try and make it accessible to everyone. We try and talk in simple language. We try and make it actionable. But again, are the people that either are not fully aware of the importance of diet on their health, not interested, or have too many other barriers where their priority might be just simply getting some sort of energy on the table, or it might be a choice of heating the house or getting energy on the table. We can't change that, sadly, at Zoe. We can do the best that we can do to advance our understanding and to give back to society. But the disparities that we have in people's nutrition education and their quality of the diets they're eating is a huge worry. And I think what drives this even further is the fact that we know that heavily processed, unhealthy food is on average about 40% cheaper than healthy, minimally processed food. Changing that is going to have a bigger impact than anything else on the whole population health.
Absolutely. A really difficult problem to solve. Well, Sarah, thank you so much for talking today.
Pleasure. It's been great to delve into loads of things, Liz.
Great talking to you, Sarah.
Bye.
Bye-bye. Hope you enjoyed the latest episode of the podcast. And if you've enjoyed the show, if you could leave a review, that would be much appreciated. It really helps. Many thanks for listening. Bye for now.